Welcome to STEM Talk.
STEM Talk. STEM Talk. STEM Talk.
STEM Talk.
STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society. Hi, I'm your host, Don Carnegas, and joining me to introduce today's podcast is a man behind the curtain, Dr. Ken Ford, IHMC's director and chairman of the Double Secret Selection Committee that selects all the guests who appear on STEM Talk.
Hello, Don. Great to be here.
So our guest today is Dr. Tommy Wood, is a UK-trained physician and an assistant professor of pediatrics at the University of Washington, who's also a visiting research scientist colleague of ours here at IHMC. In today's interview, we follow up on some research that Tommy has done since his last appearance on STEM Talk, a two-part interview that took place a little more than a year ago. In that two-part interview, episodes 110 and 111, we touched on Tommy's research into the importance of metabolic health. And since then, Tommy has published a new paper that was actually just published by the American Society for Microbiology, and it's titled Reframing Nutritional Microbiota Studies to Reflect an Inherent Metabolic Flexibility of the Human Gut, a narrative review focusing on high-fat diets.
Today, we will be talking to Tommy about this paper, as well as a new grant he just received to examine the effects of azithromycin on premature brain injury in a ferret model.
But before Ken and I get to our interview with Tommy, we have some housekeeping to take care of. First, we really appreciate all of you who have subscribed to STEM Talk, and we are especially appreciative of all the wonderful 5-star reviews. As always, the Double Secret Selection Committee has been continually and carefully reviewing iTunes, Google, Stitcher, and other podcast apps for the wittiest and most lavishly praise-filled reviews to read on STEM Talk. If you hear your review read on STEM Talk, just contact us at stemtalk@ihmc.us to claim your official STEM Talk t-shirt.
Well, Don, the poems and reviews just keep rolling in. Today we have another review written as verse and was posted by Henrik Dahlin from Sweden. The review reads, I lost 1 little pound and my wife got scared and said if you lose more pounds than me, you'd be losing your phone. I begged and pleaded with her day after day that she would start listening to the podcast my way. She gave me a kiss, and then she sent me on a walk. I told her I don't always listen to podcasts, but when I do, I listen to STEM Talk.
Well, so thank you, Henrik, and thank you to all of our other STEM Talk listeners who've helped STEM Talk become such a great success.
Okay, and now on to our interview with Dr. Tommy Wood.
STEM Talk. STEM Talk.
STEM Talk.
STEM Talk.
Hi, welcome to STEM Talk. I'm your host, Don Carnegas, and joining us today is Tommy Wood. Tommy, welcome back to the podcast.
Thanks so much for having me back. I think one of my main career goals is to establish an unassailable lead in appearances on STEM Talk by a single guest. So I very much appreciate the opportunity to come back.
That's a good goal. And also joining us is Ken Ford.
Hello, Don, and hello, Tommy.
So, Tommy, you have a new paper out just published by the American Society for Microbiology, and it's titled Reframing Nutritional Microbiota Studies to Reflect an Inherent Metabolic Flexibility of the Human Gut, a narrative review focusing on high-fat diets. So in our last interview, which aired about a year ago, you talked about the importance of insulin sensitivity and also metabolic health. And yet, as you pointed out, more than 80% of Americans have some kind of metabolic disease. In that interview, we also talked about the role and importance of fats in our diets. So we were thinking that it would be a good idea to circle back and follow up on this new research that you've done on metabolic health, the microbiome, and high-fat diets. But first, it might be good to revisit some of your key points about insulin resistance and also the importance of metabolic health, and also why so many Americans struggle with this. Can you expand on that a little bit?
Sure. So insulin resistance is essentially what we measure or can measure as an expression of the cells in your body being exposed to more nutrients than they can metabolize. And this is determined by a number of things, including total energy load, inflammation, and oxidative stress. So, for instance, when a fat cell reaches a certain size, it becomes insulin resistant because it can't store anymore. There's more energy coming in than it's able to then store. Similarly, when somebody has an infection or an autoimmune condition that comes with a large inflammatory burden, many or all of their organs may become insulin resistant because of that inflammation. It's part of the sort of inflammatory response, shifting nutrient availability around the immune system. equally, when somebody does a large volume of it or intensity of exercise, the oxidative stress generated by that exercise, if it's excessive, can cause some temporary insulin resistance as well. So there are multiple different pathways down to insulin resistance. But the reason why many people struggle with this, millions of people struggle with this, in my mind, largely comes down to our modern lifestyles. So due to the food environment and ultra food processing, we're generally in a state of caloric excess. We know that things like sleep deprivation and shift work and chronic stress can also cause insulin resistance, particularly over time. And lack of adequate movement is a big factor too. So not only is your muscle tissue your biggest buffer when it comes to normal glucose uptake and metabolism, but being sedentary is actually in itself pro-inflammatory. And on the other hand, movement or exercise is actively anti-inflammatory and over time can be antioxidant by epigenetically increasing our capacity to deal with oxidative stress. And then it's also important to mention that societal factors play a role here too, because the environment in general that we're exposed to, so everything from the quality of the air and the built spaces around us to discrimination and systemic racism, can actually affect inflammatory pathways and through epigenetic mechanisms, and then that affects insulin sensitivity long term. So in general, we struggle with insulin resistance because large swathes of the population live in environments that actively work against their well-being in multiple ways, and at the same time we're overfed with poor quality calories, we're underslept, we're overstressed, and we're undermuscled.
Doesn't sound promising.
No.
As you point out in your review, there's a common perspective in much of the nutritional microbiota literature that high-fat diets are harmful to human health, at least in part through their modulation of the gut microbiota. And there are several other studies, however, that seem to take the opposite view, and they support this notion of an inherent flexibility in the human gut, in our microbiota, with respect to its ability to adapt to a wide range of various food sources. So perhaps there's some sort of a more nuanced story here. Can you give listeners an overview of the gut microbiome and how research in the past decade has really explored the effects of the gut microbiome on metabolism, immune system, sleep, moods, and cognition? Earlier in STEM Talk, we had Alessio Fasano on one of the early episodes, and he did a terrific job on this. But if you could sort of recap this for the audience, I think that would really frame the rest of our discussion.
I have to admit that I'm not an expert on the gut microbiome, and Alessio Fasano is a fabulous researcher in the area. And this paper actually was in collaboration. One of the main authors is Dr. Lucy Mailing, who is a gut microbiota researcher, and a lot of what we wrote about was based on her body of work and synthesis of the research. So it's kind of my pleasure and honor to be able to work with somebody who's really an expert in that and learn from them. In line with your question, I think, like you alluded to, the last 10 years or so have been called the decade of the microbiome, because it wasn't really until maybe 2010 or so that this stuff really took off. However, as the field has gathered steam, people have taken stool samples from different patient populations and looked at correlations between what's in the stool, what's in the gut, and pretty much anything you might care to ask about. So multiple disease states and lots of things that I'm interested in, like neurological development and athletic performance. And the gut clearly plays a significant role here. We have a lot of both basic science research in humans, some of the basic physiology that tells us that the gut is going to influence the rest of the body and vice versa. And the gut is increasingly implicated in a number of neurodegenerative and autoimmune conditions, as well as all the other normal physiological functions that you just mentioned. However, the real problem so far is that we're very limited in what we're able to actually measure. So, for instance, when this sort of field was getting started, you might have seen a lot of papers that talked about the ratio of Firmicutes to Bacteroidetes, which are basically two phyla of bacteria, each potentially containing thousands of different bacteria. But this is what we could measure. We could just measure those phyla and look at the ratio between the two, and that ratio tended to increase in certain states, like with obesity. However, that's like taking a few square feet of seabed and taking the ratio of mollusks, which is snails and bivalves and octopuses, and a ratio of that to arthropods, which is insects and crustaceans, right? Those are two different phyla of animals, and saying that based on that ratio, we know something about the environment. And hopefully that sort of example makes people realize that there's probably that's a very blunt tool, and there's very little that we can really understand about that. And things have improved, but there's still a lot that we need to do. So part of the goal of the paper was to make the case that we shouldn't paint ourselves into these ideological corners about what's good or bad for the gut, from a dietary perspective, before we really know what it is that we're looking at.
So many of our guests on STEM Talk have observed that diet is a major factor that shapes both the composition and the function of the gut microbiota. However, much of the debate among dietary nutritionists and others focus on health effects of dietary components. For example, fiber is generally seen as not only beneficial but also necessary, while high-fat diets, and especially animal fats, are singled out as detrimental to the gut microbiota. And even high-protein, low-carb diets are targeted as playing a casual role in various cancers, cardiovascular disease, diabetes, and other disorders. So, Tommy, can you give our listeners some history on how fat, and especially high-fat diets, have become public enemy number one in many circles?
Yeah, so this is a long and controversial history, and I know you actually have had other guests who've talked about some of this probably better than I can, but people often talk about the relative contribution of researchers like Ansel Keys and some of the what we might call loose epidemiological studies that associated fat, particularly saturated fat, with cardiovascular disease risk. And then this sort of led up to, we think in the 1980s, the first dietary guidelines for Americans, which then called for the reduction of fat, and again, particularly saturated fat. And then as a result, there's some debate as to whether the population listened to that or not, and whether people did actually end up eating less fat or more carbohydrate, or whether it was just more total calories. But a lot of the problem in my mind was really compounded by essentially what now makes up our modern food environment, which is large food manufacturers who, based partly on these guidelines and partly on crops produced due to agricultural subsidies, they get to take these highly processed nutrient and protein poor ingredients that are rich in carbohydrates and relatively poor in pretty much everything else, and they're cheap to produce, and then sell them as quote unquote heart healthy, whilst also making a large profit margin. So there's a big kind of vested interest there in terms of what foods are available to people, as well as engineering them to make them easily overconsumable. But more recently, both animal studies and nutritional epidemiology studies have continued to focus on fat for other health outcomes as well, like Alzheimer's disease or certain cancers. But the data are really very dirty. And again, you guys have covered previously some of the issues with nutritional epidemiology. But when we talk about some of this stuff and we dig into it, and this is, again, another sort of theme of the paper, when we talk about something like a high-fat diet, and we're researching that in mice, a high-fat diet in mice also contains a huge amount of sugar. And when we look at high-fat dietary patterns in humans, which we do because we don't really have the data to look at individual components necessarily because the data quality is so poor. So we lump people into dietary patterns overall, which are slightly easier to study. And then we might say that this is a high-fat dietary pattern, but then we lump things together like cheese and meat with cookies and pastries, and then we assume that those things are interchangeable with respect to human health when they aren't really.
And yes, you just mentioned, as others have, some of the limitations that one sees in preclinical animal studies. You just talked about mice being fed a high-fat diet. It's often really a high-fat and high-sugar diet. And
yeah, I call it's a cheesecake diet, essentially.
And really one of the biggest confounders there is the mice are essentially low-fat consumers, high carbohydrates. So they're a bad analogy to a human eating a low-carbohydrate diet.
Yeah, a lot of this sort of research, and as a professional rodent researcher, I appreciate a lot of the potential issues here. And the two things come up, right? First, we talked about, there's the language around the construction of high-fat animal diets. But then there's also the general applicability of the rodents that we're using to human disease. And a lot of this comes down to a problem with one particular strain of mouse, which I may have mentioned previously, actually, the C57 Black 6 or Black 6. And if anybody's interested in that, there's a very nice article on the Slate written by Daniel Engber about this a few years ago. But essentially, right now, about 93% or maybe more of all animal research is done in rodents. 2/3 of that is mice, and 2/3 again of that, potentially up to 50%, is done in this one strain of mouse. And like any one strain of mouse isn't inherently good or bad for research, but we know that different strains respond very differently to different interventions that we might care about. So changes in diet or caloric restriction or anything else. So the strain that you pick will have a dominant effect on the results, which means that at minimum you need to be testing things in multiple strains, if not multiple species, before you then try and translate them to humans, which really isn't done. And we also know that the black 6 is a bit of an outlier in a few things that we might care about. So it has altered antioxidant handling that particularly affects the females. There are also outliers for certain responses to pain and neurological conditions. And in this setting, they're also particularly susceptible to weight gain and health problems on a quote-unquote high-fat diet. So we start first with a species that is adapted to or bred to respond well or thrive on a lab mouse chow diet, which is essentially low fat cardboard plus multivitamin. And then we pick the specific strain of mouse that's then going to respond badly to the diet that we're going to use. And then finally, the diet that we use, we call it a high fat diet when in fact, it is, more like a processed standard American diet that's very high in both fat and sugar. And then we do this research and then we present the results to the public as if, we have strong evidence that what happens when a black 6 mouse eats cheesecake is directly relevant to humans, which may or may not be the case. And then we also extrapolate and say, well, this is a high-fat diet, so therefore any diet that has a high proportion of fat is going to have the same quote-unquote negative effects, when in reality, those things aren't true.
Yeah, it's borderline ridiculous on the face. You tell people, follow the science. You mentioned earlier the need for a more nuanced view of fat and our microbiota's ability to adapt to a wide range of dietary sources and food sources. Could you elaborate on that a little bit? The paper was really interesting.
Thank you. And what it all really boils down to is the difference between the simple story that we're told and what actually you sort of see in practice. And the simple story is that plant-based fibers are metabolized by a healthy gut microbiota to produce short-chain fatty acids like butyrate. And then our gut epithelial tissue, so the cells that line our gut, then use butyrate to support healthy metabolism and function. And so in that story, we're told that anything that displaces those fibers in the diet, so particularly high-fat animal-based foods, that either then reduces the substrate for those quote-unquote good bacteria to produce short-chain fatty acids, or, in some versions of the story, they'll say that, those animal proteins and fats are directly damaging. So while the effect of those fibers is true, right? So we know that if we eat certain fibers, they are metabolized by gut bacteria, they produce short-chain fatty acids. This supports normal gut physiology. But in the absence of those, there's actually an increasing body of evidence that suggests there are other potential sources of energy that come from fats and proteins. So, if we take this view that, only this one type of substrate is going to fuel a healthy gut and a healthy gut microbiota, we ignore this whole other area of research that suggests that actually the gut is much more flexible than that and can metabolize a much wider range of energy sources, which I think is going to be appropriate because humans eat a wide range of foods and can thrive eating a wide range of foods.
Exactly. And part of the challenge is, you mentioned a healthy gut microbiota, but we're not really sure what that looks like. I mean, that's not entirely clear either.
Yeah, so the one of the statistics we quoted in the paper is that any two people will only share about a third of their microbiota, and two otherwise healthy people may have no overlap whatsoever. And so then, who do you pick as your example of, well, this is a healthy gut microbiota? And one obvious example that we, again, highlighted in the paper is using bifidobacteria as a genus, which are generally sold as one of the most critical components of a healthy gut microbiota. They are obligate anaerobes, they are butyrate producers, and we see them in healthy, westernized populations. However, in certain other healthy populations, like hunter-gatherers, such as the Hadza, they don't have any bifidobacteria really as part of their gut microbiota, but they're also a population which has almost no chronic disease. So, saying that bifidobacteria are critical components of a healthy gut, immediately you can see that that's not true. And it's worth mentioning that, even then, right, just because the Hadza don't have bifidobacteria doesn't mean that should be our goal either, because they exist in an environment that is very different from the one that we're in. And so this problem, defining a healthy gut microbiota is particularly the case when we go back to those blunt tools that I was talking about in terms of how we've been assessing the gut microbiota. So the majority of studies so far use what we call 16S amplicon sequencing, which only gives you details at the genus level when we know that specific functions of bacteria in the gut microbiota are happening at the species level, and then particularly at the strain level. So to give you another example, the genus level is me saying that animal is a canine, right? But you don't know if that's a wolf, a coyote, or a domestic dog, right? So you kind of narrowed it down, but there's still a lot of stuff that could be a lot of different things there. If it's the species, then right, I know that canine is a domestic dog, but I can't tell you which breed, because that's sort of analogous to the strain. And so the strain, which is, when we're looking at probiotics, when we're looking at, specific functions of specific bacteria in the gut, we really need to be looking at the strain level. And that's where I can say that's a French bulldog and not a Great Dane, which, by looking at them, there's a very different dogs with different personalities and would do different things in different environments. So we really need that level of detail, and that's not a level of detail we're getting in most studies. We also know that important functions, so say butyrate production, happens, in bacteria in very different parts of the phylogenetic tree. So even though those bacteria aren't related at all, structurally or genetically, they can still have very similar functions and benefits within the gut. So one important thing as part of future microbiota studies is going to be to focus on metabolomics, proteomics, transcriptomics, and look at function of bacteria rather than just which bacteria are present. And another issue that kind of feeds into this focus on microbiota accessible carbohydrates as the thing that is required for healthy gut function is the fact that we're looking at stool, which largely reflects what's going on in the colon, whereas it's not telling you as much about what's happening in the small bowel. And this is important because a lot of the metabolism of protein and fat is happening in the small bowel, and so important, beneficial things may be happening there, but we're not able at the moment to measure them. And again, so the fact that we're looking at stool is kind of biasing which area of the gut might be most important in terms of the microbiota. So in general, we're going to have to improve things really to understand this by getting samples from higher up in the gut, and then also having a better idea of really which bugs are there, but then also exactly what they're doing.
So Tommy, how do we go about reframing the debate about fat and high-fat diets to better reflect what seems to be the overall evidence?
This was probably my favorite part of the paper and the process of putting it together, which is the sort of under the remit of the first author, Jonathan Scholl, who's a philosopher of science and health and is trying to develop the area of philosophy of nutrition, which, as you guys probably appreciate,
good luck with that.
Yeah, exactly. But I do believe he can make some inroads. But what he lays out is the importance of the precision of language around research, and that's where, if we're going to reframe the debate, that's really where we need to start. So like we said, instead of calling the mouse high fat diet just that it's high in fat, we need to specify that it's high in sugar as well, and that it's highly refined, and then we can compare it to diets that are high in fat but aren't highly refined or high in sugar. And it's just sort of with a human example, right? We know we can take a vegan diet, and that could be made up of salads and lots of fresh fruit and vegetables, or it could be made up of French fries and Coca-Cola. And we wouldn't say that a vegan diet has a specific effect when both of those we know are going to have very different effects on our physiology. And we need to do the same with our language, when we're talking about mouse studies. And then we also need to apply the same thing to human studies. So we can't take all aspects of, say, the standard American diet and lump them together and then assume that they're all equally contributing to poor health, which is essentially what we've been doing so far. But like I mentioned, one of the problems is that the quality of the data that we have so far in nutritional epidemiology is so poor that it's almost impossible to isolate out the effect of individual nutrients, or foods. So there's a lot of work that needs to be done there in terms of improving that as well. But when we do, we really need to be able to collect the data that allows us to do that. So I think that my final point about reframing the debate is really that we need to be better at acknowledging what we do and do not know and what we need to improve on. So like we said, outside of certain randomized controlled trials, nutrition research is really very highly flawed. And what we're looking at is just a load of noise. And then when you take noise from nutritional epidemiology and you pair it to noise from gut microbiota research, then essentially, you just have no idea what you're looking at. And we need to be more honest about that.
So, Tommy, as we've been talking about, there are new technologies and a greater interest in gut health today. And in the past decade, as we've been talking about, researchers have significantly improved our understanding of gut microbes. But I also understand that you believe there's a need to better understand the human microbiota in an evolutionary context as well. Can you talk a little bit about this?
I think it's almost a bit of a cliche nowadays, but I often return to the title of a famous essay by the geneticist Theodosius Dobzhansky called "Nothing Makes Sense in Biology except in the Light of Evolution." And I think in this context, we have to remember that humans are incredibly adaptable inherently and resilient, or we should be, and this includes our guts. So as we migrated across the planet, our diets diversified hugely and at various times would have had vastly different levels of the different macronutrients and different sources of those macronutrients. So to me, it makes sense that our guts have to have the ability to adapt metabolically to whatever it has access to. So that would include plant-derived microbiota accessible carbohydrates, based on the season or geography that makes them prevalent. But in other settings, then, fat or protein-derived substrates, or, fatty acid metabolism-based substrates during periods of fasting when there is no food, your gut has to be able to adapt to those too. Or else, if we didn't always have, some Metamucil on hand, right, our guts would have fallen apart in the absence of any plants that were available. So in my mind, we may need to view nutritional microbiota research from the assumption that the gut is eminently adaptable and flexible, rather than one where it's inherently fragile and requires a very narrow range of foods to support its function.
So in your paper, you cite several times a study by Duke University's David Lawrence and his colleagues, and that study highlights how quickly and reliably the human gut microbiota adapts to dietary changes. So can you talk a little bit about the significance of this research?
You're right. We do spend a good amount of time in the paper discussing this other paper by David et al., which was in Nature in 2014. And actually, one of the figures in our paper is a reproduction of a figure from their paper. And what they did was mainly a crossover study. They had 11 participants, 10 on each diet, but 9 of them did both diets. And they spent 5 days on each of these diets, and one of them was a plant-based diet, a low-fat plant-based diet consisting of 300 grams of carbohydrates per day. They were coming from cereal, vegetables, rice, lentils, and fruit, and a lot of fiber, so 25 grams of fiber for every 1,000 calories that the participants ate. And then on the other diet, there was called the animal-based diet, which had less than 3 grams of carbohydrates per day, and 30% of the calories came from protein, 70% of the calories from fat, and it was mainly eggs, meat, and cheese, and essentially no fiber whatsoever. And that group, or the group when they were on that diet, they were in measurable ketosis by day 2. On the animal-based diet, what they saw was a decrease in bacterial populations associated with fiber metabolism, as you might expect, and an increase in bacterial populations that are bile tolerant or associated with protein metabolism, again, as you might expect. At the same time, the traditional short-chain fatty acids like butyrate and acetate decreased about 50% on the animal-based diet, although they didn't disappear. But then there was a significant increase in iso short-chain fatty acids like isovalerate and isobutyrate. And in general, when people cite this paper, they sort of use it to say, well, this animal-based low-fiber diet was bad because we saw a loss of butyrate. And that's not really how the paper sells it, nor is it really how you would align those results to the sort of evidence in general. Because for multiple reasons, in terms of the different metabolites or substrates that the gut can use for metabolism, isobutyrate, which significantly increased on the animal-based diet, can replace most, if not all, of the functions of butyrate. So what you're seeing here, rather than some detrimental effect of eliminating fiber, is you're seeing a shift in the metabolism of the gut, which is sort of normal and expected, a shift based on what's available, to a slightly different metabolism that still supports all the same functions, or at least that's the point that we made in the paper.
So, Tommy, can you give listeners an overview of the gut barrier function and its role in health and disease?
Yes,
really interesting topic.
Hopefully, yeah. And again, one that is eminently complex. But we could sort of, as an overview, we could consider maybe 4 main components. They would be the epithelial cells we've talked about a few times, and the other cells that sort of line the gut wall. There's also the tight junctions between those cells. You mentioned Alessio Fasano, who's really an expert in tight junction regulation. Then we have immune cells that are sort of surveilling the gut and outside environment. The contents of your gut being the outside compared to your body for pathogens and other things. And then we also have this mucus layer that acts as a protective component, and then also supporting both the microbiota populations as well as some of those immune components. And so each of these different components, both individually and together, have in some way been implicated when studying the impact of the gut and disease, and probably most diseases, particularly chronic diseases that we've looked at. So at the most extreme end, you might have inflammatory bowel disease, where all of these layers and functions are disrupted, and there's a resulting dysregulation in nutrient uptake, and then systemic and localized, significant inflammation, which is associated with, significant disease and poor quality of life. But then you also see it in maybe less expected places. So in TBI studies, traumatic brain injury, in both animals and humans, after the injury, you see significant gut dysfunction and an increase in gut permeability. And then at least part of the systemic immune response that comes from this damage to the gut then contributes to the severity of symptoms in the brain. All this, basically says this sort of very whistle-stop tour says that there's several complex moving parts. But in reality, we're really right at the infancy of trying to unpick all the different parts of, what's gut, what's body, what's the interaction, what are the feedback loops, and then how does that then change, with diet or disease, risk, or treatment.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research lab pioneering groundbreaking technologies aimed at leveraging and extending human cognition, perception, locomotion, and resilience.
So, Tommy, there's considerable research that supports the many therapeutic effects of the ketogenic diet, which we talk about here a lot on STEM Talk, and its effects on many aspects of human health. But what about the gut specifically?
So, as you might expect, a lot of the research that's been done on the ketogenic diet and the gut has focused on epilepsy, which is where a lot of ketogenic diet research sort of came from. And when you look at it, and when you look at the effects of the ketogenic diet in general, on the gut microbiota, again, what strikes you is the results are incredibly heterogeneous, which makes it more interesting, but then also slightly frustrating when you're trying to sort of create a simple story out of it, which at this point we probably can't do. So, like one example that we focused on in the paper was a recent study that got a lot of press actually, that looked at mouse models of epilepsy and showed that a ketogenic diet reduced the seizure burden, as you might expect, and that part of the mechanism of that was by increasing the relative abundance of a bacteria called Akkermansia muciniphila. And then, to kind of probe whether this was, actually the case, they then put animals on a non-ketogenic diet, and then they gave them this bacteria as a probiotic, and they saw the same effect. So they sort of surmised that part of the benefit of the diet was through this change in the gut microbiota, and then this specific bacterial species had anti-seizure effects. However, when you then look at other models in mice and then put them on a ketogenic diet, so there's an example where they had mouse models of autism and they put them on a ketogenic diet, which improved the behavior of the quote unquote autistic mice. That effect was partly through reducing or almost completely eliminating Akkermansia muciniphila. So in these two different models, the ketogenic diet is beneficial, but has dramatically different effects on the gut microbiota. So other important components, like we mentioned earlier, in general, we see that either ketogenic diets or exogenous ketones will reduce the number of bifidobacteria. And like I said, most people would say that having less bifidobacteria is a bad thing. But as we know, there are certainly situations where you can be very healthy and have none whatsoever. So in reality, when you look across all the different studies, and again, largely in mice, but all the different studies looking at ketogenic diets in different disease states or different disease models, how the diet affects the gut microbiota, it really depends from model to model and study to study. And then when we've looked at, and the studies are small so far, looking at the ketogenic diet and the gut microbiota in humans. So we mentioned one, the David study, which is very short term. There was a longer term study that looked at the gut microbiota in patients with multiple sclerosis on a ketogenic diet. And it wasn't particularly controlled, so it's kind of difficult to take, really hard conclusions from it. But the main thing that they showed was that these shifts in the gut microbiota as a result of the diet take a long time to sort of, there's an early change and then there's a longer term change. And so the main point being that depending on the disease, the person, other environmental factors, you will have different responses to a ketogenic diet in terms of the gut microbiota. But then those changes will also change over time. So if we're only looking at a single time point and then trying to say, well, this is a good thing or a bad thing or not, we're really going to get a very incomplete picture.
So a key point that you make in the paper is that there are multiple strands of evidence that question many of the assumptions about normal metabolic pathways in the gut. And along with short-chain fatty acids, there are several other molecules that can serve as sources of fuel for gut epithelial cells. And the idea of a preferred fuel source may be skewed from studying people and rodents who eat large amounts of microbiota-accessible carbohydrates. So you write that while butyrate production may be reduced on a ketogenic diet, other molecules can potentially take butyrate's place to help maintain the gut barrier function. So can you talk a little bit more about this, Tommy?
In general, it seems that there are 3 additional potential groups of metabolites that can support metabolism in the gut, in addition to butyrate and the other traditional short-chain fatty acids. So, like I mentioned, based on the David paper, the iso short-chain fatty acids like isobutyrate from the breakdown of protein can also provide a source of energy for gut epithelial cells. In addition to that, acyl carnitines from bile acids, so that's basically from the breakdown of fats, it's a fatty acid chain attached to a carnitine group. These can also be used as sources of energy in the epithelial cells. And finally, the ketone bodies, acetoacetate and beta-hydroxybutyrate, this time not coming from the gut, but coming from the systemic circulation, they can also be used by epithelial cells as a source of energy. And in fact, butyrate metabolism goes through ketone body metabolism in order to be used by the gut cell for energy. So it goes through the parts of the ketone body breakdown pathway that acetoacetate or beta-hydroxybutyrate could actually enter into lower down the pathway. So those pathways are identical and overlap. So you have to turn a butyrate into the intermediates in the ketone body metabolism for them to be metabolized in the first place. So the upshot of this is that the gut relies on fatty acid metabolism in general for energy, but the substrates can come from both inside and outside the gut. And, those will be available in differing proportions and from different sources depending on, macronutrients or, relative intake or type of the diet. So even though, there's all this, it's basically a final common pathway, it's all based on fatty acid metabolism, but there's multiple different sources depending on what the person is eating. So this is what we mean in the title of the paper by saying the gut is inherently metabolically flexible.
Makes sense.
Mm-hmm.
Have there been any studies that have assessed the effects of ketones or a ketogenic diet on the gut barrier function we talked about earlier, explicitly on barrier function as opposed to gut energy?
So as it's phrased that way, we couldn't find any specific studies looking at gut barrier function on a ketogenic diet, at least at the time of writing the paper. But there was one interesting study that we talked about, which looked at ketone body metabolism in gut epithelial cells. And in gut intestinal stem cells, they seem to have all the machinery to produce ketones, and having that machinery is actually necessary for normal responses to injury and repair. And then when you look at more mature cells, they don't have the machinery to make ketones, but they have all the machinery to utilize ketones like we talked about. So, there's definitely an idea that in the setting of certain injury or inflammatory states, particularly because in those states you might alter or have impaired butyrate uptake, which you might see, say, in inflammatory bowel disease, then in that setting, potentially providing ketone bodies as a source of energy will then also help with repair as well as supporting normal healthy metabolism in gut epithelial cells.
In a sense, you can think not a direct effect, but an indirect effect due to what you're eliminating when you're eating a ketogenic diet. So you're not eating the very things that open up the tight junctions typically.
Yeah, absolutely.
So in 2015, the World Health Organization classified red meat and processed meat as probable carcinogens. And then the International Cancer Microbiome Consortium, however, released a statement in 2019 that said that there's a need for better human studies into how the microbiota influences carcinogenesis. So, Tommy, we're curious if you can shed some light on what's going on in terms of research into high-fat diets and any potential link with cancer.
This is a particular area of interest for Jonathan, which spends a lot of his time focusing on diet and cancer. But I think the picture, both that we outline in the paper and in general in the research, is very much in line with what we've discussed earlier, which is that for many of these questions we simply don't know. And the WHO classification of meat as a probable carcinogen was based on some really quite poor quality data. If there's any signal there, it's from dietary patterns that include a lot of poor quality and highly processed meat and all the things that go with that. And even then, when you try and look at the effect of processed meat, the absolute risk increase is pretty small. multiple people have now basically said what I think is true, which is that we don't have strong data either way. However, we can certainly say that a high-fat diet in the sense of the standard American diet is a significant contributor to metabolic disease and then the downstream diseases, which includes many cancers. However, we have to reiterate that this does not automatically apply to whole food-based diets that include a significant proportion of their calories coming from fat or meat.
I hate that term, standard American diet or Western diet. It's sort of self-loathing. You go to the Middle East and it's certainly not a standard American diet. And the situation with diabetes is much worse than here. And, I think it's just an unhealthy diet or a modern diet, maybe.
Yeah. The finding a way to describe it that encompasses all the terrible aspects of it is still something that I struggle with. Yeah.
So what about some of the mouse models and preclinical studies that show ketogenic diets or ketones are cancer suppressive?
Yeah, I think that's an important point and something that the audience of STEM Talk will no doubt be familiar with. And just like in multiple areas of cancer research, there are studies to suggest that a ketogenic diet might be beneficial for colorectal cancer, both by reducing glucose availability, but then also more directly by altering, say, redox status within the cell, which may then increase apoptosis and death of tumorous cells. So while we're obviously still waiting for formal clinical trials, I think it's definitely an area of interest. And then, like I mentioned earlier, if we zoom back up to conditions that might predispose to colorectal cancer, like inflammatory bowel disease, there's certainly multiple lines of evidence that may say keto bodies could be actively support the regeneration and the metabolism of a healthy gut, as well as, those that say maybe anti-inflammatory and some other effects. So if you're trying to prevent colorectal cancer in the first place, there's certainly multiple lines of evidence that say a ketogenic diet may be beneficial in those who are at risk.
You acknowledge in your paper that we have been discussing that there are various objections to this notion concerning the effects of fat and protein on our gut microbiota, and you address 3 of these objections in the paper. Could you just briefly go over those and elaborate just a little on them?
We obviously wanted to make sure that we covered, at least to some degree, what are likely to be major objections to the idea that a high-fat diet can be beneficial or at least not detrimental to the health of the gut. And 3 main ones that come up again and again are LPS, endotoxins, TMAO, and bile acids. So we covered these each sort of briefly. And LPS or lipopolysaccharides, these are gram-negative bacterial endotoxins that kind of sit in the bacterial cell wall. And there's lots of this in your gut because you have lots of gram-negative bacteria in your gut. And that's perfectly normal. However, there's a correlation and some very nice preclinical studies between circulating endotoxins, chronic inflammation, and metabolic disease or insulin resistance. We also know that fat increases the translocation of LPS across the gut. So in studies where they're trying to look at LPS circulating after a meal they might use or they have used to increase the amount of circulating endotoxin in humans is drinking some heavy cream, because there's lots of fat and that brings a lot of LPS across the gut wall. However, what it really seems like is that the main issue is reduced barrier function and not the normal processing of LPS uptake, which is that LPS gets taken up into chylomicrons as part of fat uptake from the gut. Those chylomicrons end up in the liver, and then the LPS is actually sort of metabolized and broken down there. And that's sort of like the normal pathway and seems to be beneficial. Whereas if you have breakdown of gut barrier function, which you might have in the setting of insulin resistance or chronic inflammation, then you get this sort of unregulated uptake of LPS across the gut wall, and that's probably much more detrimental. So having more circulating LPS in the setting of metabolic disease is probably a consequence rather than a cause necessarily. We also talk about the fact that not all LPSs are bad. Many are beneficially immunomodulatory. So part of the the function of the immune system is to know what is and isn't worth targeting. And there seems to be some crosstalk between what's in the gut and the immune system to say, hey, we aren't the bad guys, don't go after us. And that some of that happens through certain LPSs. So overall, we felt that probably wasn't a reason to say that, a high-fat diet is necessarily going to be bad. The next one, TMAO, people may have heard of. It's produced in the metabolism of choline and carnitine, so obviously prevalent in animal-based foods. And over time, these have been associated with cardiovascular disease risk, although the evidence really is quite weak. So it's used to say that, eggs and meat are bad for cardiovascular disease because TMAO is associated with cardiovascular disease. But a lot of seafood dramatically increases TMAO, and seafood generally isn't associated with cardiovascular disease. And not when you look at the data in a meaningful way, I don't think meat or eggs are either, just to make that point. But the source of the choline probably matters too. So there's been a very recent study that looked at eggs versus isolated choline as a supplement. And so eggs, as part of a whole food, didn't increase TMAO, but a supplemental choline did. And then when you actually look at the gut microbiota, the bacteria that are more prevalent on animal-based diets, these sort of like bile-tolerant, protein-breaking down bacteria, they may actually contribute to the breakdown of TMAO. So they may be beneficial in this setting, which is normal again. You sort of like have an overall shift in the gut microbiota in response to that kind of diet. And then sort of finally, there's a possibility that TMAO is essentially a big fat red herring anyway, because some recent Mendelian randomization studies have suggested that there's actually reverse causality going on. So it's metabolic disease and insulin resistance causing increases in circulating TMAO, as well as contributing to cardiovascular disease rather than the TMAO being the culprit. So again, there isn't really strong evidence that this is an area of concern. The final one, bile acids, as you might expect, bile acids contribute to the breakdown and metabolism of fat in the diet. So their presence in the gut increases with increasing fat intake. And if you do sort of isolated studies in the lab with very high doses of bile acids, then there's some evidence that they may be damaging to the cells in the gut. However, at physiological doses, they actually seem to be more beneficial. And like we mentioned, acyl carnitines from bile can actually, in and of themselves, be metabolized by the gut epithelial cells to support normal, healthy gut metabolism. So again, as with all of these potential objections, we think that overall, on balance, these aren't reasons to say that a high-fat diet is going to be bad for the gut.
So, Tommy, you've done a really good job of making the point that we need to reframe research when it comes to investigating nutritional microbiota research, that high-fat diets are harmful to human health. So, in your mind, what questions need to be asked or answered when it comes to nutritional microbiota research, and also how should studies be carried out?
I think at this point, pretty much everything is still up for grabs. And I would make the point again that in most scenarios, we don't really know yet what it is that we're looking at. Mm-hmm. So I don't really think I could say these are specific questions that need to be asked and answered, because probably most of them still need to be asked and answered. And so the important points would be, making sure that we're precise in our language and being honest about what we do and don't know. And the majority of it is stuff that we don't know currently. And then on top of that, I think the lenses of metabolic flexibility and evolutionary biology are going to be really important as well.
So, Tommy, you gave a talk back in early 2020 at IHMC about nourishing the human brain, and I think the title of your lecture was Brain Health Across the Lifespan. I understand that you recently received a large grant to examine the effects of azithromycin on premature brain injury in a ferret model. So first of all, huge congratulations on the grant.
Thank you.
Yeah, but next, can you give us some insight into what you'll be studying?
Sure. So if people have heard me speak on STEM Talk previously, they'll have heard me talking about the ferret as a model for brain injury, particularly in neonates, but then also in adults as well. And over the last few years, my lab has been developing models in the ferret of premature brain injury, and particularly important because the areas of the brain that are damaged in babies born prematurely, particularly extremely premature infants, so those are born before 28 weeks of gestation, those areas of the brain aren't very well represented or easily modeled in rodent models. And so if you want to do some studies to better understand what's happening there, then we think that the ferret is a nice way to do that. So we developed this model, and now we're in a pretty good place to understand how that brain responds to injury and how it responds to therapies. So this grant is to look at azithromycin as a potential therapy in this model, which again models this sort of extremely premature infant population. And there's some data from a collaborator of ours on this grant, John Barks at the University of Michigan, and he's tried it in a rodent model of neonatal brain injury, of sort of more later term brain injury. And so now we're in a really good spot, and I think we have the right model to then look at this in extremely premature babies. So there's a combination of things that we'll be doing. There'll be some in vitro slice culture work, there'll be some in vivo work in the model, and then we'll do lots of behavioral testing, MRIs, particularly focusing on the effects of azithromycin on the microglia in the brain after injury.
And as you said, you'll be focusing, you're just talking about azithromycin as a neuroprotective agent in your upcoming research. So what is it about azithromycin that you find particularly exciting?
So this grant particularly focused on, or they asked for proposals that were focused on repurposing of drugs for conditions affecting pregnant women and infants. And so that's one of the reasons why we focused on azithromycin. And it is cheap. It's FDA approved. It's shelf stable. It can be used in any environment, including in low and middle income settings. We also know that it's safe in pregnant women and neonates. And like I kind of alluded to, one of its most interesting functions is rather not as being, so people may or may not know, azithromycin is an antibiotic. It's a Z-Pak if anybody's taken that for, say, pneumonia. But at the same time, it has this effect on microglia, which are these immune cells in the brain. I mentioned them, I think, previously on our last podcast, and I also talked about them briefly in the IHMC talk that I gave. And they're important, sort of, again, the main immune cells in the brain, but after certain injuries, they can be chronically activated and then contribute to ongoing inflammation and neurodegeneration in the brain. And azithromycin seems to modulate microglia in order to essentially send them back to their more sort of resting, either normal resting or more anti-inflammatory states. And there's multiple like spinal cord injury and other brain injury models where azithromycin has been shown to be neuroprotective. But this is really the first time that I think that anybody's applying it to this extremely preterm group. And another reason why this could be really useful is because extremely preterm infants are generally exposed to a huge number of antibiotics when they're being treated in the neonatal intensive care unit. So if you're going to give them antibiotics anyway, and you can give them an antibiotic which also may have some beneficial effects on the brain, that's really, two birds with one stone. And then sort of beyond that, because we know the importance of microglia and chronic microglial activation in certain neurodegenerative conditions, like for me personally, I'm also very excited that azithromycin might be useful in the treatment of those at risk of dementia, sort of at older ages.
Well, that was really interesting, Tommy. And one of the neat things about this grant is it will facilitate you actively collaborating with your wife, Dr. Elizabeth Nance, who was our guest on episode 71. And she's also a faculty member at the University of Washington. Can you tell us a little bit more about how each of your work sort of intersects on this particular project?
Absolutely. This is probably the biggest. We do have some other grants that we work on together, but this is probably the biggest one that we'll work on, at least so far. And as listeners may know, Elizabeth's work, she has a significant expertise, particularly in nanoparticles and imaging of the brain, but then also as part of that, culturing slices of live brain tissue from multiple species, rodents, humans, primates, and now ferrets, so that you can have a better look at what's happening, in real time. If you have, in an in vivo model, we call it sort of like a whole animal model. Yes, we can look at, certain time points. We might be able to do an MRI scan, or, maybe we're going to look at pathology in the brain, but that's sort of like a single, there's usually only a single time point there. You have to sacrifice the animal in order to get that. However, when you use brain slices, you retain a lot of the structure and function of the brain, but you can really track it over time, and you can sort of image deep parts of the brain and different cell types in the brain, in real time as they respond to, say, an injury or treatment, say with azithromycin. And this is something that Elizabeth's lab has significant expertise in, and this is the first part of the grant will be working in vivo in ferret slice culture. And like I alluded to, a lot of the focus is going to be on microglial cells, again, an area that Elizabeth has a lot of experience with. And this is something that we've been developing over the last few years, again, largely in her lab. But we hopefully have a paper that we're about to submit that looked at microglial responses to injury in a ferret slice culture model. And then based on how the microglia in a different region of the brain respond to the injury, we can then predict whether that region of the brain will respond to a given treatment. And we've tried multiple treatments, including acetoacetate, those who are interested in ketones, and erythropoietin, which is a neuroprotective agent that's being used in babies currently, or at least in clinical trials. So we have an idea that in this setting, we can track microglial changes and then use that to determine or predict which areas of the brain will be most injured and or may respond to a given therapy. And I think it makes sense that not all areas of the brain are going to respond similarly to the same injury or the same treatment. And particularly if we're thinking about acute brain injuries, traumatic brain injuries, or strokes, or cardiac arrests, or the premature type brain injury that that we're studying here, it may be that you need multiple therapies at the same time in order to treat all areas of the brain that are affected by the injury. And that's something that we're really hoping to dig into over the next few years.
It's really exciting work, and we really enjoyed interviewing Elizabeth before. So how is she doing otherwise?
She's doing very well. She says hello to everybody. So a few months ago, she found out that she was approved for tenure, which is very exciting, obviously. And over the past year, she was promoted to associate chair in the department for undergraduate programming. So she's basically in charge of all undergraduate teaching now in chemical engineering at the UW. And this is at the same time, obviously, running a large research program. So, obviously doing very well. A superstar, as always, though I think after this past year, we're probably both ready for a bit of vacation. Hopefully that comes soon.
Like you deserve it. Both deserve it.
Yeah, well deserved.
Yeah.
Finally, with respect to your interest in brain health across the lifespan, could you talk to us about how your work in neuroprotection for premature babies really might inform things that could be relevant for adults looking to optimize their long-term brain health?
This has become a real hot topic in the past year. I think I touched upon it at the beginning of my talk at IHMC, now nearly, I guess it was 15 or 16 months ago. And then in the past year, sort of the research, or at least the interest in this has really exploded. And what we're seeing increasingly is that in babies born prematurely, they have more advanced brain aging based on various machine learning algorithms that you can apply to MRI scans. And the more premature you were and the sicker you were as a baby born prematurely, basically the bigger difference between your brain's biological age and your chronological age. We also see the same thing in those who have either progressive mild cognitive impairment or frank Alzheimer's disease or Alzheimer's dementia, that their brains age more rapidly than their chronological age progresses. So there's now increasing evidence that says that this trajectory of adults born preterm of brain aging is going to result in accelerated or increased risk of Alzheimer's disease or cognitive decline. And we're already seeing in this population, who are only just now reaching their 50s and 60s, the first adults who are born, very preterm. They're only just getting to their 50s and 60s now, but they already have an increased risk of all-cause mortality, as well as a number of metabolic and other chronic diseases. And so, this is really starting, even before you're born, the in utero environment, but before that is basically creating a trajectory of brain aging and other aging and risk for chronic disease that's going to continue throughout your entire lifespan. However, I think it's what's one of the things that's really interesting to me is that I think all adults can learn then from some of the protective aspects that contribute to neurodevelopment or protect against the effects of early life brain injury in babies who are born prematurely, for example. So what's nice about the research in that area is obviously people are very interested in making sure that babies, regardless of what happened early in life, have the best outcomes later in life. And a lot of this then translates also to people who were not born prematurely, but want to protect cognitive function late into life. So the environment is really critical. And I mentioned in my talk, and is relevant again here, that both development of the brain and decline of the brain are driven by demand. What are you actually asking your brain to do? And babies in a more challenging learning environment with more opportunities to learn and fail, and then learn from those failures, they do better cognitively. We also know that babies born preterm have better executive function as children if they have better motor skills. And so when you're looking at the effects of exercise on the developing brain, it seems like developing motor skills and challenging the motor system has a significant effect on other parts of executive function as well. And then we see the same thing in adults. So in meta-analyses looking at effects of exercise, those that include a balance component have a greater effect. So yoga or dance or things of that nature, rather than static, either endurance or resistance training. And one of the reasons I think that is because challenging the vestibular system is a much more significant driver of neuroplasticity than other ways of challenging the system. Because if you challenge your balance, that's a significant challenge to your overall survival. So it's a greater stimulus for the brain because, there's more of a need then to respond to those inputs. So doing things that challenge the motor system, the vestibular system and balance, they're important for babies, and they're also more important to try and create new neuroplasticity within the brain pretty much at any age. So what I think we're seeing more and more of is that not only is this trajectory something that you can look at across the entire lifespan and is affected by things across the entire lifespan, but at each stage, things that are important, for development are also important for preventing decline.
It's really interesting, Tommy. So, summing everything up, you've accomplished quite a lot in your career in a relatively short time. But I understand that one of your most important goals in life, and by the way, I'm just going to say this is besides making the most appearances on STEM Talk, which you talked about earlier.
Is Tommy in the lead? I think he is.
I think he is.
I think he just took the lead.
Yeah, I think with this one, he takes the lead. But one of your other most important goals in life is to be the best, and I quote, dog dad as possible. So how are those two boxer pups of yours doing?
They're doing great. And actually, a skill they've developed is to know when I'm starting to wrap up recording a podcast, and then they like sit outside the door waiting for me to be done, which is really cute. But they are eating a pure raw meat based diet full of meat and bones and organs and stuff that I make by hand, and they're loving every second of it. And it's good for them, and maybe it's good for humans too.
Yeah, lucky dogs, right? You see, most dogs eat this weird dog chow that looks like nothing they needed those teeth to eat.
Yeah, exactly. And our dogs certainly wouldn't eat that again.
They would turn their nose up.
Yeah, not after the diet you feed them. Well, Tommy, it's been fantastic having you back on STEM Talk. Thank you so much for joining us today.
Thanks so much for having me again. This is a huge pleasure, as always.
It was fun, Tommy. Thanks.
STEM Talk. STEM Talk.
STEM Talk.
STEM Talk.
So, Ken, I've always thought that good science is driven by curiosity, and that's one of the things that I most admire about Tommy. He's so curious about the world and science, and he's constantly asking questions that really steer him into so many different realms of research, and today's interview is just another great example of that.
Yes, Dawn. As Tommy pointed out, there's a common belief, actually it's dogma, that high-fat diets are harmful to human health, at least in part through their modulation of the gut microbiota. We need people in science to constantly challenge the conventional wisdom, which, as in the case of high-fat diets, often gets treated like gospel and has more confidence attached to it than is warranted. Tommy did an excellent job today of painting a more nuanced view of the inherent flexibility of the human gut and our microbiota's ability to adapt to a wide variety of food sources.
If you enjoyed this interview as much as Ken and I did, we invite you to visit the STEM Talk webpage where you can find the show notes for this and other episodes at stemtalk.us. This is Dawn Carnegie signing off for now.
And this is Ken Ford saying goodbye until we meet again on STEM Talk.
Thank you for listening to STEM Talk. We want this podcast to be discovered by others. So please take a minute to go to iTunes to rate the podcast and perhaps even write a review. More information about this and other episodes can be found at our website, stemtalk.us. There, you can also find more information about the guests we interview.